everybody. Welcome to our day 2 of the Masterworks program. It's my pleasure to welcome you all here today, and as is our tradition at Island Pacific, we'd like to recognize the ancestral and unceded territory of the Squamish Nation, upon which both the school and this chapel that we're in here today reside. A little bit of lesson for you. Last night while I was on YouTube checking out our stream, up popped a YouTube channel that told me how to pronounce in Squamish Bowen Island. So given that we're talking languages coming up here, I'm going to try to say Bowen Island in Squamish. Nuch lelachwum. So we are on Nuch lelachwum's unceded and ancestral territory. So I would like now to welcome the master of Masterworks, Jennifer Hendrickson, to set the stage for us on our exciting day 2. Please, Jen, and welcome up here.
Happy birthday.
Happy birthday. Happy birthday. Sorrowing, death, and dying. Happy birthday. The big 5-0.
Thank you. So glad that we are live streaming all of this also, so the whole world will know that I turned 50 today. So, thank you for coming to Masterworks on our second day. This is a really big day, big week for our grade 9s because this is the culmination, the jewel in the crown of their 4 years at IPS. And they've watched Masterworks since they were in grade 6. And have thought about what they're going to study for the entire year. If they choose well, they're pretty excited about it, and the research that we've gathered is that a lot of kids, actually what they study in grade 9 when they're 14 goes on to what they actually do as a job or study in university. So it's really quite interesting. It's sort of like a great stepping stone. So our students have written a 15 to 40 page paper. And today they're going to show you the gems of that paper and what they've learned. They've also been mentored by an internal advisor or faculty on staff and also an external advisor. And we're super grateful for those advisors that volunteer their time to help our kids open their eyes to whatever it is that they're usually professionals in their field. So it's quite amazing the great mentorship that we have relationships that have formed. So there are a few conventions for this week. We call the grade 9s by their last names. And also this statue was donated by Ian Henley. It's called Taking Flight. This is the bird of wisdom and a student hanging onto it, taking flight and flying off into whatever their adventures are going to be. So we are live streaming. And there's a question period after the presentations. Anyone who is out there in the virtual world can type in a question to youtube@go.islandpacific.org, and we'll read them out during question period. I'm going to give you the rundown of the other presentations today before we start our first one. At 9 o'clock, Ms. Jada Madeline Scott will be talking about teaching French as a second language. At 10 o'clock, Hunter Stephanie Thompson will be talking about the future of medicine. At 11 o'clock, Rose Willis will be talking about making decisions. At 1 o'clock, Tobin Fail will be talking about wolves in North America. And at 2 o'clock, Colin McLean will be talking about catching light evolutions in chemistry. So, without further ado, I would like to ask Miss Mademoiselle Madeline to join us today to talk about teaching. Mademoiselle Madeline, can you come to the podium, please?
Sorry for being late, guys. My bad. Okay. Sorry, my staff meeting went a bit overboard. Okay, I'm here, so hi guys. I'm Jade Madeline, and I'm your substitute for today. And I'm supposed to be covering for Carrie, who claims to be sick. Today we're going to be going over teaching, or just basically how to teach languages, French in particular. So today we are going to be covering learning styles, the teaching challenges that you will face and how to overcome them, tips and tricks on how to make your French classes fun, and yes, this is possible. And then I'm going to share my experiences with teaching, which is not to be judged by anyone. Okay? By the end of this lesson, you'll understand this fundamental principle of teacher training, which is education works best when it is entertaining. I've always loved languages, but most of my peers do not, and I've always wondered how teachers can get languages into the heads of stubborn teenagers who don't want to learn. I was inspired to do this project by some excellent French teachers I've had over the years, and now I'm going to inspire you. Okay, please take out a sheet of loose leaf. We are going to be beginning with the learning styles. Okay, so basically the first thing that you guys have to know about each person is that they have their own way of learning things that is unique to them, and it's developed biologically and developmentally. Now, VARK. VARK is the acronym that describes the 4 main learner types. So visual. V for visual. These people learn best when information is presented to them in the form of pretty pictures, brochures, and colorful squiggly things on graphs. They can easily get distracted by pretty things out of the window. Georgia, hi, up here. Jeez. Okay. A for auditory learners. These kinds of people are fans of audiobooks, music, and group discussions and lectures. If you're an auditory learner, you're probably listening to me right now and staring blankly at my slides. R is for read-write learners. These learners absorb the information best when it is written in lists, diaries, textbooks, and well-constructed slideshows. You're welcome. And finally, K is for kinesthetic learners, the most challenging type of learner for teachers. These people learn best by trial and error and figuring out how things work as they go, like experiments, demonstrations, and hands-on activities. So these people are probably dying in their seats right now. So you have a class with a mixture of students with various learning styles, so what do you do with them? One of the different ways teachers deal with classrooms filled with different types of learners is the technique of differentiation. Differentiation is matching teaching to the needs of each learner. Teachers have different ways of using differentiation. They break down complex concepts to make them easier to understand. Another way is allowing students to choose how they present their work in line with their learning style, using a hook to capture a student's interest, usually by tricking you guys into listening. Fun, right? And lastly, creating an inclusive environment by looking at differences as assets instead of weaknesses, like that kid who knows all of the Turkish swear words. Could be helpful. Now we are going to talk about our problems. Don't worry, I won't complain for too long. To figure out what problems teachers have, I sent out a survey to a bunch of them and asked what their main struggles are with keeping you guys engaged. So we have students with varied levels of ability in one class, the students who don't really care and don't see why they're learning French in the first place, and Google Translate, every student's best friend. Keeping students engaged is also a struggle, especially when cell phones aren't allowed in class. Also, Leela, hi. Are your phones more interesting than this presentation? Apparently not. You can put them at the front of the desk here and you can collect them at the end of the class. Okay, just to keep things fair, I also surveyed the IPS students to find out what they find most boring about French learning. Students don't like grammar and verb conjugation tests, working by themselves or silently, doing worksheets, taking notes, or having a bored teacher. So how do we meet the students halfway? Some teachers try to challenge their students, use interactive activities that incorporate students' interest. They try to use games and videos, think of interesting projects and assessments, and try to be the funnest version of themselves, which can be positively exhausting. Okay, so let's put this all into practice, shall we? The process of lesson planning is a very personal experience, and every teacher has a unique way of making their own lesson plan. After asking multiple teachers, I found out that most of them do not make lesson plans for every class. Carrie. If you look inside of the planner of an experienced teacher, which I have on multiple occasions, when they're out of the room, of course, you'll find a few notes for a rough outline of what will happen in each class. Of course, this is no help when you're just starting out, so I'm going to share with you the basic method of structuring a lesson. The key elements that you will need to cover are your lesson objectives, how you're going to deliver them, how you plan on assessing the students, and the materials you need. Now, let's flesh this out. You're going to have to warm up your students so that to get their brains at least functioning slightly before presenting the information. You would have to go through various phases of practices and check-ins. Make sure to leave some time at the end for questions and review. Always leave a bit of wiggle room at the end of your lesson, just for the unexpected. Like that time Scott ended up talking about how deep a lake was for the entire class. Yeah, that actually happened. Now, we're going to learn about lessons from my lessons. Now, to be very, very, very clear, this is a no judgment zone, okay? Okay. I taught a class to the grade 6s and one to the grade 7s. Now, grade 6. This class has a total of 11 students, but only 7 of them take French class. Since I would not have many students to handle, I thought that this class would be as easy as pie. I constructed a very impressive looking plan for teaching them some animal vocabulary in French. As you can see, I had all of the key elements, and then I fleshed out how I was going to deliver each piece of information, and I even used the learning styles to create the teaching methods. I had a total class time of 1 hour and planned how much time each thing would take. Very organized. Okay, so Carrie started off the class and then handed it over to me. I was definitely nervous and had to stop my hands from shaking. I began the lesson by asking the students to write down as many animals as they knew in French so we could have like a mini competition to see who knew the most. Luckily, the whole student body knew roughly 3 animals in total, so thank you, students. That idea went down the drain. I then asked each student to read out the animals that they knew, and I wrote them down on the board. Fortunately, I am terrible at spelling, so I couldn't spell most of the animals that they were listing, and I should have made fun of myself for not being able to spell words, but I did not because I felt useless and embarrassed. So far, so good, am I right? The students had been very creative with their knowledge of animals by only knowing the animals in French that sounded like rude words. Let me show you. Le faucon, le phoque, et le coq. Yeah, thank you, kids. I did not know whether I should laugh or think it was funny or gently tell them off. I did neither and carried on. I then taught them new vocabulary and asked the students to write down the words on a piece of paper. I tried to write the words in large letters on the board to make them easy for the students to read. But my writing was all over the place, as you can see. One of the students even asked Carrie if one of my R's was a V. Okay, no. Let's take a poll. Who thinks that this thing right here, serpent, looks like an R? Raise your hand. Come on. Okay, who thinks it looks like a V? You all are blind. Okay. Next, I introduced je préfère. I taught the students how to say I prefer a dog or whichever animal that they chose. And then I went around and asked each student if they prefer one animal over the other. I then said to the class that we were going to play a mini game that involves a slideshow. It was at that very moment that I realized I'd completely forgotten to make the slideshow. I was so nervous about teaching the class that I completely forgot to make it. I panicked when I realized this and made it in class while Carrie taught something else. After I finished making the slideshow, we played the game, and it wasn't too bad. The students were excited about the game, and the teams were tied at the end of class. Danae and Rose are now going to come up and quickly give you a mini taste of what the game was. Let's just tap this chair. Okay, let's go. Okay, ready, guys? Go. Danae.
Je préfère l'oiseau.
Marie, okay, 1 point to Danae.
Je préfère le poisson.
Bravo. Okay, thank you guys. Bye-bye. Okay, and then just to top it off, I didn't have any time for my concluding section. Okay, so now, how did that lesson go? Well, let's start off with everything went wrong. I was not able to get through all of the vocabulary that I wanted to, not because it was taking too long, but because even I was getting bored. So I stopped myself from torturing the kids more. I forgot to make the slideshow, so I had to carry on as if it was deliberate not to have the slideshow ready. Grade 6 students, I want to congratulate you for surviving that class. I wasn't confident in my approach, and I didn't own the space. So in conclusion, my first class was a nightmare. Round 2. I plan to teach a 45-minute class on French food to the grade 7s. The grade 7 class has 18 students in total, which I thought would be harder to teach than the grade 6 class because there were so many more students. I began planning the lesson and I started using the table that I used last time. Then I remembered what that lesson was. So I decided to make up my own lesson plan rather than following any templates. I wrote the plan out in a very traditional manner on paper. I based my lesson around these 2 pages from Carrie's textbook that described 10 traditional French foods. I wrote down what I would read and what I would ask a volunteer to read. I also scripted what I was going to say for each after somebody read each of the passages. I numbered everything so I wouldn't get lost, and I even put it into my own words. Like here, I didn't know what capers were, so I wrote that they were apparently nasty. Okay. Carrie started the lesson and presented a mini slideshow of her weekend before handing the class over to me. When I went up to the desk to start teaching, one of the students told me not to be nervous. So I said that I wasn't nervous. What a lie. I handed out the reading sheet and sat in front of the class. I read out the title and then the passage and asked the students what some of the words meant, just to make sure everyone understood. I joked with the students and told them that I had to look up one of the words because I didn't know what one of them meant in French or in English. Gastronomy. Okay, no. Put up your hand if you're under 18. Okay, put up your hand if, keep your hand up. Keep your hand up. Now, keep your hand up if you know what gastronomy is. Rose, you want to take a guess?
Oh.
Okay. Anybody else? Indra?
Is it it's like salami, but it's not.
Not really. Okay, moving on. Once there were no more volunteers for reading, I chose random students, especially those who were being a bit disruptive. I asked if anyone had tried some of the foods we were talking about, and we had many discussions around that. At this point, I handed out the worksheet and explained the bare minimum about it, and then let them loose. I went around answering questions and checking in on people until the end of class. Okay, here's a mini video of me teaching. So. kept my cool, joked around a bit, and tried to make the students feel as comfortable as possible. Everyone understood what was going on, and I think I kept them entertained and on their toes. I learned a lot about what not to do after my first class with the grade sixes. For example, I learned to be relaxed and not embarrassed when I didn't know a particular word in French. I just casually asked Carrie, and when she didn't know, I looked it up online. This is a great way to model that it is okay not to know every single word in French and how to use the correct resources to understand unfamiliar vocabulary. Not Google Translate. I think I could have explained the worksheet a bit better. Most of the students didn't know how to do one or two of the questions on the sheet because they didn't know what was required of them. Also, one of the students didn't want to read in front of the class, and I feel that it pushed the student a little too much before moving on to a different person. At least now I know when to back off. I found this class a lot easier to manage than the grade 6 class, even though there were so many more students. Some of the grade 7s were a bit rowdy and Carrie had to speak to them, but they were mostly fine. I was nervous before this class, but I felt quite confident by the end. I really enjoyed teaching the class. It was a lot of fun. The students were enthusiastic and they were able to do the worksheet that I'd given them. Some of the students even came up to me after the class and told me that I was a good teacher. They were so kind, and I think we all had fun doing this lesson. Anyway, another important part of teaching is assessing your students. Okay, assessments. Another thing that you need to know about your class to make it a success is to assess them without boring them to death. I made a worksheet for the grade 7s, a quiz for the grade 7s, and then an official test for my Madre. But before I started making any sort of assessment, I had to do some research of my own. There are 3 types of assessments. Assessment for learning determines what students already know and what they need to learn. Assessment as learning is like a check-in with the students as the students learn. This could be in the form of a simple quiz, a brief conversation with the whole class, or any piece of work that isn't marked, kind of like a formative assessment. And assessment of learning is the one that students hate the most. Assessment of learning determines students' final level of achievement and typically involves a marked project, test, or exam. This is the graded work that appears as a mark on the students' final report cards. Structure of an assessment. When creating a quiz for a French class on vocabulary, conjugation, or grammar, it is important to use different types of questions, starting with the easier questions first to warm up the students before moving on to harder ones. Also, make sure to cater for the different learning styles. Now, the grade 7 worksheet. I made the grade 7 worksheet for the French class to accompany the Commons French food class that I taught. It had 3 sections with questions progressing from easiest to hardest. I added pictures as well, which was not a smart move because it took forever to line them up on the page. Carrie thought that the worksheet was not long enough, so she added another page of questions. She was right. By the end of the class, the faster students had already finished the worksheet. I didn't know how long it would take them to complete the worksheet, but at least now I know for next time. I created a quiz for the grade 7 class to test them on the present tense conjugation of ER verbs, which they were learning with Carrie at the time. I included an invisible table, which took forever to remove the border lines to make it invisible. So I'm not going to do that next time. Writing sentences and paragraphs for the fill-in-the-blank sections was difficult and time-consuming because I had to make up sentences using verbs and words students are familiar with, which also had to make sense. Otherwise, students could get caught up on words that they didn't know and become overwhelmed. I also created an extension activity as an extra challenge for the students, so it took a lot longer to make the quiz than I had expected. Carrie gave the quiz to the students and I marked the responses the next weekend. It took me a little while to mark all 15 of the quizzes. Does anybody want to have a guess of how long it actually took me? Indra?
1 hour, 20 minutes.
Yeah, it took me just over 2 hours and it was crazy. The marking took forever. I didn't know what to write on the paper sometimes, especially when the student hadn't done so well. I couldn't really write good job on a score on the paper with a final score of 17 out of 50. I also didn't feel comfortable writing feedback because I'm not an actual teacher. Calculating the scores was the worst. I realized that I had miscalculated the total number of questions on the quiz only after I'd marked 12 out of 15 of them. So I had to go back, recalculate, and erase the previous marks, which I had stupidly written in pen. Then there was the matter of the bonus questions, where students had to write 3 sentences of their own. I had no idea how to mark them, and the grammar was wrong, but the conjugation was right, or vice versa. One student completely threw my marking system when they correctly conjugated verbs that don't exist. Smart kid, but what was I supposed to do with that? Also, what do you do when a student gives you multiple answers for a question, only one of which is correct? In these cases, I gave the students half marks. When I was about halfway through the quizzes, my brain started to hurt. How do teachers cope with marking different quizzes for multiple grades? Okay, moving on. I'm actually proud of this one, so don't be judgy. Okay, over the spring break, I was supposed to go on vacation to France with my parents. My mom was going to try and advance her French learning as well. Since we were not able to go because of COVID-19, I tried teaching my mom instead. I taught her how to conjugate all of the present tense verbs and designed a test for her. It was a good test, if I do say so myself. The first thing I did was change my font to Century Gothic, which is a very, very nice font and makes me happy. It took me around an hour and a half to make the test and about 5 minutes to mark it. And yes, there was only one student, which definitely made it faster. Learning from experience, I wrote the total number of points that you could get for each section on the test, which made my life easier when marking it. All I had to do was write the number of questions she answered correctly. There was a set of questions that required my mom to translate a few simple sentences into French, which I set up so she would get 1 set of points for correct grammar and an additional set of points for correct verb conjugation. There were 5 of these questions, so the maximum points that she could have earned was 10. This way, there was no confusion about how to mark that section. It took my mom around 30 minutes to complete the test, and she did very well. My mom said that the test was challenging but interesting and doable. This means that my teaching was successful and that the test was pitched at the right level. Okay, I was very happy with the marking and the overall outcome of this test. Jeez, okay, I'm running out of time here, so let's recap. During this lesson, students, you have learned about the 4 learning styles and how they should be incorporated into lessons. You have learned that learning styles are your friend in the classroom, and you have learned how to make a lesson plan and how to create an assessment. It turns out that what you see in the classroom is just the tip of the iceberg in terms of the work that teachers have to do. Behind the scenes, there are hours upon hours of creating assessments, planning lessons and units, and evaluating their methods, not to mention the bucket load of fun marking. Teaching is not easy. If you're afraid of public speaking or acting, do not take this job. You are basically performing in front of judgy students every single day. Teaching is also scary, but for some it is exhilarating. At first, I was petrified of teaching, but after a few successful experiences, I felt more confident. It made me feel really good to help students learn, and I'm looking forward to getting that feeling again when I become a teacher one day. And that concludes our lesson. So girls, you can collect your phones on your way out. But first, I have a few people I would like to acknowledge. First, I would like to thank my faculty advisor, Carrie Marantet, for meeting with me almost every week and setting many due dates for me to make sure I actually did stuff. Half the time I didn't, but you never really found that out. I would also like to thank my external advisors, Laura Meager and Caroline Schraff, for meeting with me every submission and looking over my work as I progressed. You guys are the reason why I was inspired to do this. I was inspired to do this project. Thank you. Richard Scott, thank you for reading my paper and taking out the extra V's, T-H-E's, out of my work. And Scott, thank you for being my manager, and thank you for being my guinea pig as well. Without you, my paper would not look like a professional or anything that looks remotely like a report. And I have no idea how you constructed some of those graph things. So, thanks, Mom. I would also like to say a huge thanks to Jen for reading my very long paper and managing to stay awake the whole time. I don't know how you read everyone's paper without giving up halfway through. Thanks for not giving up on us, Jen. Okay, for some reason, every kid who does Masterworks thanks their class, but honestly, I have no idea why. You guys didn't do anything. But I guess you struggled with me, so thanks, guys. Okay, and lastly, I would like to thank Garfield for keeping me sane while constructing this slideshow. Thank you.
Masterworks experience. I'm so proud of all the courage you've shown through this whole process. So great job. So you've acquired so much knowledge about what teachers do and the challenges that they face in the classroom on a day-to-day basis. How do you think this information is going to help you as you move forward in your education and in your careers as in high school and so on? Well,
usually when you start off teaching, right, you do a couple of practices and you fail the first time. Well, I've already done that, so I think I'm off to a pretty good start of how to move on, and I know what to include and stuff, and you know, it's just going to be easier now that I know all of the learning styles and all of the challenges. So yeah.
Excellent. So I have a couple questions from your external advisors as well who couldn't be here today, but I know Laura's watching from Switzerland and Caroline's watching on the live stream as well. So Caroline wants to know first, following your experience of teaching to different age groups and also prepared a test for your mom, which age group do you think you'd rather teach French to later in life? Would you consider teaching to very young children in an immersion school, or as a middle school or high school teacher, or even to adults in university?
I think I like this age group. They're kind of annoying and it's kind of great. So, you know, I don't know. I would find teaching adults would be kind of scary. And young kids, I don't know, after a while, kind of wears you out. So, you know, yeah.
Okay. Caroline would also like to know, have you figured out why you are attracted to teach French specifically? Is it connected to a personal experience in your life?
Well, over the years I've had many, many good French teachers, and I've just been inspired by them. And I don't know, it's just, I love French. I love the language, and I love speaking it, and I really look forward to teaching it one day.
This is a beautiful language. So Laura would like to know, Jada, you list many difficulties teachers have in teaching French, including the fact that many students aren't interested in the subject and don't see its relevance. Were there any moments during your research or when you were teaching when you questioned whether or not you still wanted to pursue a career in teaching French? What do you think motivates you to continue on this path?
Okay, well, I definitely had some roadblocks, and I definitely reconsidered teaching. I don't know, it just like, especially after that first class, I kind of was like, do I really want to be doing this? But I don't know, just the risks, kind of like the whole like good stuff that you get out of it just outweighs the risks. So.
Excellent, thank you.
Mademoiselle Madeline, I really like that you just said that, because I was going to ask you, you've talked a lot about where you failed and what was really challenging and how hard it was. So I want to know, is lot and it's hard every day. But what do you get out of it? Why would you want to be a teacher?
It's just that feeling. You just get a feeling when you're up there and teaching, and I don't know, like I feel most comfortable when I'm going around and helping people while they're doing a worksheet or something. And you know, it's just that feeling that you're kind of helping people, you know, and it's just a good feeling that you go home at the end of the day just going, cool.
I know that feeling. Okay, questions from the floor.
Jen, sorry, just
go ahead.
quickly. I think my mom wants to ask me a question. I really do.
I thought you'd learnt about backing off, yeah?
I guess it's genetic.
Yes, clearly. Okay, so I have probably two questions. The first one is, we know that, well, I know from knowing you, that you're an auditory learner, a bit of visual thrown in there. I'm sure your teachers know you can sometimes be a little disruptive. You like talking to people. Maybe it's the auditory thing, I don't know, you like to hear yourself talk. But also you pick up things quickly. So how would you teach yourself? What would you do to engage yourself? What would the lesson plan look like?
I would probably just get somebody to talk at me, and then I would have to respond. Like they're not allowed to keep on talking. I have to talk to you. But probably talking. Like Caroline and I talk a lot, as you know, and that really helped me get the language into my head. So and the pronunciation and stuff. So.
And following on from that, now that you've been on the other side of the lectern, so to speak, as a student, do you see teachers differently now from how you did before?
Definitely. I will never ask a teacher like when I'm going to get a test back ever again. Ever. No. And every single time I watch somebody teaching, it's kind of like a little note taking in my head. So, you know, just are they doing this? Are they moving around? Or are they? And I watch the kids as well if they're getting kind of bored and looking out and going, when is this going to be over? But like, I just watch stuff and it's just a completely different view, and I'm noticing quite a few different things. So, yeah.
Can I ask you, Ms. Mandel, have you always been observant about that, or is it just when you started your Masterworks and started really thinking that maybe I'd like to be a teacher that you started noticing?
I tried to notice how teachers, I always try and notice how teachers like present stuff and how they keep them engaged, but it was really after I taught that first class that I realized, okay, step back a bit. Yeah, because it was just crazy and I had no idea what I was doing, so yeah.
Ms. Jen.
Yes. Great presentation, Mrs. Madeline. I was just wondering, when you're teaching French or a second language in general, do you think it's best to speak mostly in that second language or mostly in English, or like what do you think the ratio should be?
Well, I gotta say for auditory learners, if in class, if the class, like some of the people who are auditory learners, I think that they would really benefit from that, but some people don't. I think they would pick it up, everybody would pick it up faster if they were kind of forced to speak it. So I think it would be really good to be able to speak in French the whole time.
Anna, go ahead.
Great job, Miss Madeline. I must say that you were perfect when you taught us that class. I didn't notice anything wrong. But I do have one question. If you had to name your most favorite part of teaching and your least favorite part, what would they be?
Okay, favorite part would probably be going around and helping people while they're doing their worksheet, because that makes the pressures kind of off me. And worst favorite thing, worst part. Probably reading in front of the class. I hate reading in front of people. It kind of hurts me. So,
yeah. Mr. Stringfellow, go ahead.
Very good job, Ms. Madeline. Besides French, if you had to teach anything, what would it be?
definitely math.
Can I just spark a question on that one? Do you think what you've learned in this about teaching French can apply to teaching math?
Well, I'm going to find out next year when I do this project again at Carson. So I'm going to do it the same project, just with math instead.
Really?
So
Your personal project you'll do with math?
yeah, so it's basically the same, but I'll let you know.
All right, Mr. Sokol-Snyder.
Good job, Ms. Madeline. So you mentioned how teaching a smaller groups in class and teaching a bigger groups in class was different. So do you think in the long run you want just a small class like an RPS class or like a big like public household like 30 plus kids?
Really depends on the kids in there. If they're super rowdy, don't think I would want to be in that class. But I mean, 18 is a good number, I think.
Ms. Willis, go ahead.
Excellent job, Ms. Madeline. I was just wondering, how do you think teaching methods vary depending on age groups?
I think there's a lot more fun and like acting kind of involved with the lower grades, like grade 6 and earlier. You'll have to do a lot more actions and show bigger pictures because they get distracted easily. So, and older kids can deal with lectures better, so you can rant at them for like an hour and they'll still listen to you.
Scott, can you read some questions from our virtual audience?
Will do. First question from David Jan. Great engaging presentation, Ms. Scott. I see you learned a lot about teaching. My question is, do you think a student's age makes a difference to which learning style, aka BARC, could make learning easier? In other words, does the visual style make more sense to younger students learning French?
Well, it's up to the person, really. It's the person has their own specific type of way they learn, right? It's all unique. So unless it changes over time, it doesn't really change. It's the whole, showing more pictures and giving more games and stuff when you're younger and more lectures when you're older. But it's really just how you are, and that's how you cover it. It's better just to cover the whole learning styles when you teach instead of just focusing on one. So.
Thank you. From the grade 7 students in the think tank, what is your favorite type of learner to teach?
Probably auditory. Yeah, I think so, because then I can talk at them.
And do you see yourself becoming a teacher in the future?
I definitely do, yes.
Okay. And I have one from Ted here. Wow, you have no idea just how important and insightful this presentation has been. The background research you did, plus the interviews and your actual teaching, allowed you to make a number of very powerful observations about the challenge of education from the point of view of teachers and students. Some particular gems. Teaching plans often do not work. Learning to not be embarrassed when you do not know something. The pitfalls of student assessment, don't get me started. And the time it takes to mark. I'm going to incorporate some of what you taught us today into my own ongoing exploration of how we can make schools better. I think your presentation should be required viewing for all educators. Nicely done. Dr. Ted Spear, thank you. And from the Sokol-Snyder family, wonderful, funny, and wise presentation. As someone who has experienced online learning over the last few months, do you have any thoughts on good practices for teaching online?
I think it's definitely going to be it's definitely more challenging. I guess, just being very specific, in terms of making assessments, you're going to have to make sure that all of the learning styles are definitely incorporated into that project, because then the students are more likely to put their effort into it and actually do it, because I know that from personal experience, you tend to avoid work at home. So it would be good if you had a lesson or a project that definitely incorporates all of the learning styles, so you can really focus on it. So.
One more if I can. This is from Rosie. I love this one. What advice would you give students now on how to make teachers' jobs easier?
Don't ask them to mark your quiz faster.
Nice. Thank you.
That was great, Ms. Madeline. I think you have to go to staff meeting now. So we will be having a 10-minute break and be back here for Mr. Hunter Stephanie Thompson at 10 o'clock.
glad that it's done.
Then you have less good questions. You nervous?
I'm Phil, and John to give you like a question, send in a question to Scott, or like create something.
That's the best.
So they're clear.
Yeah.
Are you everyone's gonna have like slides? Ladies and gentlemen, are you guys almost ready?
Yes.
Okay. Gentlemen of the technological world, are you almost ready? Yeah. Oh geez, let's turn this on. I'm gonna leave that open. Okay, quick. All right, load. Hey, Adam, you good? Okay. Hello, everyone. Welcome back to Masterworks of 2020. It's a unique one. It's a unique one because of COVID-19, obviously. We've got the grade 9 class and some of the grade 7 class here in the chapel, but we are also live on our YouTube channel. So thanks for joining us virtually or today in this lovely chapel. So this week is a huge one for the grade 9s. They have been working on a topic of their choosing through the entire year, and they've written a 15 to 40 page paper. In this case, it was 40. And they've been helped along on this journey. They've been mentored by a faculty advisor within the school and also external advisors who are professionals in their field. So we're super appreciative of the time and efforts put in by essentially volunteers to help our students learn so much about their topic that they've chosen. So today we are lucky enough to get the gems of their research, and they get to become the teachers today. And with that, we just had a teacher, and now we're going to have Professor Dr. H.S. Thompson join us today to talk about biotechnology.
Where will we be in 50 years? What will we have to worry about? 500 years ago, we might not have been as lucky as to live past, say, 30. And you know what? The methods for even dealing with the disease back then were not quite pleasant, might I say. Like bloodletting, which is just not very nice. We don't really have to worry about dying from diseases anymore, such as polio or influenza. And even for someone with a deadly disease, we have methods to extend their life expectancy. Medicine is always making giant leaps, and in the future, we may not even need to worry about dying whatsoever. Welcome to Module 1.4 of my Biotechnology course. I'm your professor, Dr. Thompson, and today we'll be looking into biotechnology used in clinical medicine. I hope you all did the reading, as there will be a test tomorrow, 50 multiple choice, 50 word answer, 50 paragraph answers, of course. Firstly, I'd like to ask, Mr. Weeb, to you over there in the corner, what are the targets of biotechnology?
DNA, RNA, and protein.
Looks like someone's been doing their reading. For those of you who haven't done the reading or, haven't even bothered, like some kids, today's lecture should enlighten you. There it is. Oh, how unfortunate.
Come on, Professor.
Technology is not my strong suit.
Just good? Yeah.
Yeah. Biotechnology used in healthcare today is a promising area of science, utilizing protein, DNA, and RNA, said as Mr. Weep so kindly, and as well as protein transcription and translation for diagnostic or treatment-related therapies. These products are developed by a host of genetic engineering techniques and methodologies specifically directed towards the physiological modification of cellular end products and or processes. Although biotechnology is providing extremely powerful tools capable of altering the very basis of life, for public good, of course, numerous ethical questions of wielding such power are also being raised. biotechnology techniques being used in clinical therapies are enhancing the results of current treatments and have the potential to alter the pathway other risky treatments take. Biotechnology poses a significant primary treatment against genetic disorders such as cystic fibrosis, infectious viruses, or types of cancer by either inhibiting the expression of dysfunctional genes, tackling the process that certain viruses take, or by preventing cellular mutations and mistakes made in cell replication. On a more pathogenic-based lens, biotechnology can be used to harness the already existing characteristics and processes of bacteria and viruses, and even mimicking them in order to understand and utilize their pre-existing characteristics. An example of this can be found in the well-known CRISPR. This blob of an enzyme came from the all-feared bacteria. When bacteria are attacked by a bacteriophage, they capture snippets of the DNA and put them into something called a CRISPR array. This is so that the next time the bacteria is attacked, it can produce an RNA version of that viral DNA. They then use Cas9 to disassemble the invading DNA. This just goes to show that an understanding of genes through biotechnological and therapeutic means is the key to treating and eliminating diseases, whether they be viral, genetic, or bacterial. The purpose of this module is to A, provide insight into current and future biotechnology, and B, how it serves to assist in therapeutic treatments against inborn or pathogenic-based diseases and processes. Unlocking the full potential of our genes is a key to ending generations of genetic disorders and correcting mutations and mistakes made by ourselves. Genes hold the data for all living organisms, I hope you've heard, and contain the coding used for the certain creation of proteins, enzymes, and other secretions and products, all necessary for the survival of the species. To understand the importance of these genetic codes, scientists need to know the damage they can cause when mistakes are made to properly understand how we can reverse it, or better yet, prevent it. Genetic disorder is a genetic mistake caused by one or more abnormalities in the human genome. There are multiple types of genetic disorders, such as mutations caused in one gene, which is a monogenic disorder, mutations in multiple genes, multifactorial inheritance disorder, mutations in the mitochondrial DNA inherited from your mother, which is maternal inheritance, or by damage to the structure of entire chromosomes. A genetic disorder can be caused by environmental impacts, such as UV radiation, or by inheritable factors, such as recessive gene expression, mutations in the genes, or by genomic imprinting. These mutations, caused by mitotic and environmental events, can lead to permanent damage to genomic data. And you know what? Even though they look like pieces of knotted spaghetti, genes are really important because they'll hold the instruction for the key of making proteins and enzymes for both unicellular and multicellular organisms. Just the slightest change in the DNA of an organism could have massive effects, like having a switch that once flipped causes a genetic disorder to occur. A monogenic disorder is the result of a single mutated gene. There are over 6,000 human diseases caused by the mutation in a single gene. These disorders can be passed down through generations and have been shown to influence inheritance patterns. Such monogenic disorders include cystic fibrosis, sickle cell anemia, Fragile X syndrome, Duchenne muscular dystrophy, and Huntington's disease. Cystic fibrosis is an inborn disease that primarily causes damage to the lungs, but also the kidneys, liver, and the majority of organs in the digestive system. Cystic fibrosis affects cells that produce mucus, sweat, and digestive acids, and products required to digest food properly. In order for a person to develop the disorder, they must have 2 copies of the defective gene, one from each parent. If they were only to have one, they'd simply be a carrier, meaning they couldn't actually have the disease, but they could still pass it off to their children, which isn't very nice, but... The disorder is caused by the expression of mutations in both parent copies of the CFTR, cystic fibrosis transmembrane conductance regulator gene. A healthy CFTR gene provides a CFTR protein for organ membrane cells that create mucus, saliva, sweat, and other important digestive enzymes for organs in the body. For patients with cystic fibrosis, secretions that are meant to be thin become thick. This lifelong disorder can be tested by a sweat test, which measures the sweat viscosity of the individual. The most common mutation that leads to cystic fibrosis is the deletion of the 508th base in the CFTR gene, as shown in the image, which leads to the occurrence of a frame shift mutation. So frame shift mutation causes bases in a gene to shift, affecting how they'll later be read, which could have massive effects. Gene therapy has a number of techniques, and one of the most promising is CRISPR-Cas9 prime. Both of these methodologies, gene therapy and CRISPR, are related in various ways, but vary in their efficacy and accuracy in what you use them for. Biotechnology approaches cystic fibrosis by means of gene therapy. Gene therapy is a genetic editing therapy that relies on a viral vector to insert new genetic information into the patient's cells. The healthy DNA sequence will be placed into the infected gene beside the mutated DNA with the aim to express the healthy sequence instead and start to create the proper CFTR proteins again. The production of these proteins will allow the endothelial cells to manage mucus and prevent a buildup of thick fluid. You can just appreciate my image right there for a second. Very nice image. Gene therapy is an experimental technique used by scientists to correct defective or mutated genes responsible for the development of disease. It is used to add genes, replace genes, or turn off specific genes that can cause cancer, different genetic disorders, or genetic diseases. Some gene therapy trials have used viruses to correct genes by adding unmutated versions of the same gene to the patient. Gene therapy has had major success in laboratory experiments and has the potential to provide future cures for monogenic disorders such as cystic fibrosis, or even Duchenne muscular dystrophy, Huntington's disease, and other disorders such as hemophilia A, severe combined immunodeficiency, and possibly even types of cancer. However, there still are a few speed bumps to overcome with the use of viral vectors. The body's natural immune response sometimes impedes the virus from reaching its target cell and sometimes causes complications for the virus. Some viruses, such as gamma retroviruses, add genes at seemingly random locations in the host chromosomes that can disturb other cellular functions and may lead to cancer. Adeno-associated virus-based vectors, big name, but they're also much safer because they always deploy genes in the same area of the chromosome, with exceptions in various diseases such as Alzheimer's. Gene therapy relies on viral vectors to vary the genomic data to the specified spot on the genome, and scientists do this by using 4 main viruses in gene therapy: adenoviruses, adeno-associated based viruses, retroviruses, and lentiviruses. Scientists are also studying the use of stem cells and liposomes in therapy. Stem cells could learn to fight certain diseases in a lab, and liposomes could be used as a certain different form of vector to ferry the new genetic information. Gene therapy could possibly strengthen the capabilities of the immune system by correcting genes that are responsible for the failure of development of white blood cell manufacturing regions, which in turn are responsible for our innate and adaptive immune systems. I should be done there. CRISPR-Cas9 prime is the most powerful tool in terms of genetic editing to date, and also poses as a great tool for correcting gene disorders, and possibly even cancer, by accurately replacing, altering, or adding DNA sequences. There are 3 tools used by CRISPR-Cas9. They include a guide RNA, reverse transcriptase enzyme, and a Cas9 nuclease. The guide RNA is used to guide new DNA, hence the name, to its selected location in the genome. The guide RNA also contains the new nucleotides to replace a faulty DNA sequence. A hobbled Cas9 enzyme cuts one end of the targeted DNA sequence, but not both, hence why it's called a hobbled DNA sequence, to ensure the DNA machinery notices a change. The next enzyme, which is fused to the Cas9, reverse transcriptase, takes the new RNA nucleotides carried by the guide RNA and creates a copy in DNA, which replaces the old nucleotides at the site. And if you can read that, that's pretty impressive. I need my glasses for that. Because one end of the new DNA sequence was left undone, the cell's machinery comes into play and either mends the 2 broken ends or takes a random nucleotide floating around the cytoplasm and places it as a sort of connector piece, which is kind of lazy. But anyways, another option is for scientists to add another nucleotide segment for the cell's machinery to grab onto and put into place for the scientist. Because of a small mismatch in the DNA sequences, the prime editor nicks the end of the sequence, which prompts the cell machinery to finish the sequence based off of the new DNA strand. Infectious pathogens such as human immunodeficiency virus, aka HIV, remain problematic for our immune system and our immune cells. HIV is a retrovirus that specifically attacks and degrades the human immune system, allowing other small diseases to easily overwhelm the immune system, finally leading to acquired immunodeficiency syndrome, aka AIDS. Current pharmaceutical treatments use highly active antiretroviral therapy, known more commonly as HAART. HAART is an antiretroviral drug cocktail that helps to inhibit the functioning of retroviral enzymes that are critical to the virus's reproduction. This treatment allows the human immune system to repair the damage done by the virus and prevent further infection. Now, biotechnology approaches this with monoclonal antibodies, as I'm sure you've heard of sometime. The specific monoclonal antibody, 3BNC117, inhibits the HIV from infecting the CD4 helper T cells by binding to a piece of the HIV spike proteins. So you see up there those green mushroom-looking guys? Those are what the antibodies are going for, and they're also what attach to the CD4 cell. It's very effective for HIV-positive patients in combination with HAART and monoclonal antibodies together, and this could increase the efficacy rate of treatment of HIV together. Okay, I hope I haven't lost anyone. I swore my last class pulled the fire alarm on purpose. As you may recall from our last module, when we discussed the immune response, we talked about antibodies and antigens. Antibodies are tiny molecular Y-shaped proteins that are produced by the immune system to search for specific antigens, very specific. They attach to these antigens and then broadcast its location to more specialized cells that either engulf it or bombard them with molecular chemicals, which is pretty cool. Antigens are foreign viruses, toxins, or substances that cause the immune system to manufacture these antibodies in the first place. Monoclonal antibodies have two main parts. The orange variable region on the antibody is what recognizes a specific piece of the spike protein of HIV, in this case the green mushrooms, and the blue constant region is what is visible to the immune system and is what triggers an immune response. These tiny proteins are created in a laboratory by hybridoma cells. Hybridoma cells are created by fusing immortal melanoma cells and natural antibody-producing B cells. Hybridomas create mass amounts of a specific type of monoclonal antibody for therapeutic research and use. Monoclonal antibody binding targets are all identical in that they all target a very specific piece of a very specific antigen, unlike polyclonal antibodies. Millions are created by a single cell to bind to viral antigens, thereby inhibiting them from spreading and infecting. These antibodies form a clump of viruses, quite a mess, and they can easily be identified and then engulfed or destroyed by immune cells, such as macrophages. It's like a very messy puzzle that then gets destroyed. It's very messy. Monoclonal antibodies are being utilized in many current treatments and therapies targeting cancer, rheumatoid arthritis, cardiovascular disease, autoimmune disease, multiple sclerosis, viral spike proteins, and many more diseases because of their proficiency and accuracy in targeting and broadcasting very specific antigens. Monoclonal antibodies used as a treatment for infectious pathogens cannot be fully supported due to their efficacy rate dropping over long-term treatment periods. And this is because the immune system is starting to look at them and they're like, who are these guys? Like, these are kind of weird. What are they doing? So they just start attacking them. That's why HAART and monoclonal antibodies, they want to use them at the same time to reduce the resistance of the immune response. Cancer is one of the most difficult diseases to treat. Normal cells develop mutations in genes, often involved with the regulation of the normal cell cycle, to become precancerous. These activated precancerous cells undergo further mutations and evolve to dodge the attempts of our immune system to remove these errant cells. What we know as cancer is the summation of all of these mutations in the genomes of our cells that have led to the process of tumorigenesis. Further mutations can enable these tumors to metastasize and invade other tissues and organs. Cancerous cells inhibit the function and growth of natural cells and interfere with natural body functions. Cell mutations can occur because of mistakes made in the synthesis of DNA or from environmental impacts such as UV radiation. Cell mutations can lead to cancers such as leukemia and lymphomas that can sometimes be passed on through your genes. Cancer can be incredibly difficult to treat because different types of cancer respond differently to treatments, as they are composed of different people's mutated DNA. and cancers are notorious for learning how to evade certain drugs. A promising new approach to blood cancers is CAR T-cell therapy. CAR T-cell therapy is a clinical therapy that involves removing the patient's cytotoxic CD8 T cells, the assassins of the immune system, also known as natural killer cells. And what they do is they insert new genes into these T cells for a new receptor called a chimeric antigen receptor, or a CAR. These newly enhanced T cells are cultured and reinserted back into the patient and can now target, attack, and kill specific cancer cells. The therapy adds an additional receptor to the cell surface that specifically deals with lymphoma or leukemia cancer types. And while this therapy has potential, it currently remains very expensive, unsafe, and risky. But a benefit to the therapy is that the CARs, chimeric antigen receptors, cause a permanent T cell adaptation, which may come in handy if the cancer somehow comes back. As research into genetics furthers, scientists have recognized that not all carriers of genetic disorders are bad. And this is when it gets interesting, if you're not already interested. For instance, sickle cell anemia, which is a recessive disorder. If both parents were to pass on the genetic mutation, it would result in a genetic disorder occurring. But if only one parent passed on the mutation, the result would lead to the resistance of the malaria parasite. This suggests scientists take a careful approach to genome editing tools such as CRISPR and gene therapy because of the possibility of eliminating a potentially useful adaptation or mutation. Antibiotics. Okay. You see those pills in your house? Maybe your doctor prescribed them, or like your dog ate them off the carpet, which is like not ideal. Antibiotics are a natural or human-made drug capable of destroying or interrupting bacterial reproduction, meaning against life. They are currently seen as the most effective agent used against infection. They may seem like little pills, but they certainly pack a punch against bacteria. However, though, of course there's always a however, they have become less and less effective. Anytime an antibiotic is used, it risks the chance of the bacteria gaining some form of resistance against the antibiotic. As antibiotic resistance is becoming more and more prominent, we are running out of types of antibiotics to fight bacteria, and then our major treatment will become no longer useful. Kind of sad. These guys are the absolute worst, and I don't think many of you have heard of them. They're like super soldiers that evolved to specifically give you a very hard time. Superbugs are a strain of bacteria that are resistant to human-made drugs and antibiotics. They can become immune to multiple types of antibiotics and can even mix with other species of bacteria, which is just way more fun, through something called horizontal gene transfer, which I'll explain later, to become even more dangerous and incurable, or possibly more contagious and spreadable, which is lovely. The reason why superbugs are so dangerous is because even the most simple bacterial infections can become incurable, and then they can be fatal. Okay, Mycobacterium tuberculosis, depicted here, these little long elongated jelly beans, are the world's most deadly superbugs to date. This specific bacteria alone contains a waxy self-surface made of something called mycolic acid. This bacteria alone causes 1.7 million deaths every year, as it has evolved to evade antibiotics and drug-related therapies. The main reason why it's so dangerous is it hides in the host cells like a stealthy assassin, which is kind of unfair, until it decides that there are enough others of itself to attack, which I'll talk about later. Horizontal gene transfer, also known as lateral gene transfer, is a natural phenomenon used to transfer genes between multicellular or unicellular organisms. Instead of genetic information being transferred from parent to offspring, hence vertical gene transfer, genes get transferred directly from one organism to another, like me giving Pam my height. This is an important factor in the evolution of organisms. HGT is also a major factor that leads to antibiotic resistance among bacteria, and it also leads to bacteria becoming more virulent. HGT is also a major factor that leads to antibiotic resistance among bacteria, as I just said. Bacteria containing certain genes responsible for antibiotic resistance can transfer their genes to other species of bacteria through mechanisms called transformation, transduction, and conjugation. In transformation, DNA floating around in the environment is picked up and fused with the bacteria's own DNA, which is so sloppy and weird, but Transduction occurs when a bacteriophage, or a phage, accidentally inserts the resistant gene for an antibiotic or drug directly into the bacteria. And conjugation is when cells exchange DNA via sexual pillows. The processes can arm yet another bacteria with immunity against that certain compound. And even though this may seem harmless, bacteria reproduce at an exponential rate and can be passed on to different people quite easily. Considering what I just said, if conditions are favorable, bacteria such as E. coli can reproduce every 20 minutes, which translates to one bacteria producing 8 others in an hour, which translate to 2,097,152 more E. coli in just 7 hours. And in 24 hours, it's 2 to the power of 72, but that broke my calculator, so can't show you that. Which is like a lot. Like, I can't even count that high. That means if a single bacteria's genome mutates to become resistant, then in like no time, there'll just be like an army of annoying little midgets. Very annoying. Because of this mass reproduction rate, scientists are struggling to slow the spread of antibiotic resistance, because not only do bacteria spread their resistant genes to their offspring, but also to other species of bacteria. Because bacteria can genetically alter themselves, they can sometimes become more dangerous branch of bacteria called a superbug. And fun fact, did you know, before 1936, 30% of all death in the world was caused by bacteria, which is just not good. Bacteria are a menace to the medical world for a few reasons. They're difficult to find cures for because they just keep reproducing, and they can alter their DNA by pure chance. Here's the good news though, there are a few ways to cure bacterial infections. One way is to inhibit the growth of the infection or by directly killing the bacteria themselves. Scientists can do so by attacking the bacterial cell wall, or interfering with bacterial reproduction, or blocking protein production in bacteria, or even RNA synthesis. Without the use of the cell wall, bacteria cannot keep out unwanted visitors such as phages and destructive chemicals. Bacteriophages are a natural form of virus that are specific, very specific, to one species of bacteria. They attach to the outside of the bacteria and insert their viral genome into the bacteria's genome. Bacteriophage's genome effectively inhibits bacteria from replicating, producing toxins, and functioning only to produce more of itself. To prevent bacteria remembering their DNA, which I talked about in CRISPR arrays in the beginning, scientists can select certain bases and certain nucleotides of the phage's DNA to alter that won't affect the phage's creation. The therapy is very selective in the bacteria it attacks and helps to effectively reduce infection and spread and side effects. The treatment is very successful compared to antibiotics and has a very low rate of the bacteria developing any form of resistance. Phages are extremely specific to one species of bacteria and do not attack healthy bacteria and healthy human cells. Phages, shown above, insert their DNA into the bacterium, and then when their DNA is copied, it is made into different parts of the bacteriophages, which are then constructed and then released, destroying the bacteria in the process. This leads to them becoming a promising method to reducing bacterial resistance and possibly eradicating harmful bacteria and their resistant genes. Plus, they look like the Apollo space capsule, which is pretty cool. Bacteria are one of the world's most primitive organisms, if you didn't already know. They're single-celled microscopic animals that thrive in incredibly diverse environments, such as the Sahara Desert, the depths of the Marianas Trench, and inside other organisms. Bacteria are one of the supporting columns of all life because they assist in other organisms' functions and survival. The average human carries about 30 trillion human cells, which is like a lot, and carries about 100 trillion bacterial cells, which is like a lot. In terms of DNA, there are approximately 30,000 human genes, a considerable amount, and 100 times more bacterial genes, which is like a very considerable amount. Keep in mind that a single bacteria contains only like a few genes, and capable of its characteristics and functions. Bacteria play a role in every aspect of your life as they help you to digest food and eat parasitic organisms off of your skin, providing us with nutrients and many other things crucial for our survival. As bacteria can be helpful, they can also be extremely harmful to humans and other organisms. But How are microscopic organisms so small, capable of taking down larger organisms such as a human or an elephant? Bacteria have their own language called quorum sensing. This language allows them to count how many other bacteria of the same species there are by using special chemical molecules like hormones. These chemicals are produced by the bacteria and are received by other bacteria with the receptor on their surface specific to that one hormone chemical. Bacteria use this tool to count their numbers as a species before launching an attack on its host. Or they can use it to determine if they need to reproduce or take out a certain task in its host in a beneficial way. And they can use it to figure out if they need to break down protein in the digestive system or other helpful traits. This form of quorum sensing can be referred to as intraspecies communication, so within a species communicating. But not only can bacteria communicate within their own species, but they can also talk to other species of bacteria. All bacteria have a broad spectrum chemical and receptor that allows this to happen. Bacteria can communicate through this all-inclusive language to know how many other bacteria there are of specific species, to understand what it can work to do in the digestive tract of organisms. This form of quorum sensing can be referred to as interspecies communication. So, if bacteria can talk to each other within its subspecies or within all bacteria, and this communication allows bacteria to know when it should release toxins and attack its host, doesn't that mean that we could either inhibit the production of these communicative chemicals or just block the receptor? This new idea can not only help to stop a specific species of bacterial infection, but it could also be used as a broad-spectrum form of antibiotic. Scientists are currently looking into bioactive compounds used as quorum sensing inhibitors and reducers of bacterial gene expression. Bioactive compounds such as itaconamides, lycopene, and indole are chemicals found in small amounts in plants and certain foods, such as nuts and vegetables. That could be used to block quorum sensing. So when your parents tell you to eat your veggies, I definitely suggest you listen. Biotechnology is an extremely useful set of tools because of its potential for curing infectious pathogens, viral and bacterial, types of cancer, and genetic disorders. Biotechnology today is being used to genetically edit cells in order to prevent disorders. Editing technologies are also equipping immune cells to better handle cancerous cells and viruses. Although these therapies and treatments come with good intentions, they also raise numerous ethical questions towards the morality of enhancing the human species beyond evolution. As genetic editing and therapeutic technologies improve patient survival and overall health, these techniques bring with them moral concerns towards the fact that we are playing with evolution itself, and mistakes could cause generations of trauma or create a new species of pathogens, which is just not ideal. Because of our understanding of genes, we are coming closer and closer towards the elimination of diseases and disorders, though we are still just scratching the surface of all science. Biotechnology has shown that understanding the function of genes is the key to eliminating diseases, whether they be genetic, viral, or bacterial. This is because genes can help us enhance cells to better attack pathogens. Therapies can edit mutated cells and genes, and viral vectors carrying genetic material can help inhibit bacterial reproduction and function. In the future, we can look forward to genetically tailored treatments based on an understanding of our genetic makeup, lifestyle, and environment. As genes define how even disorders and cancer cells fluctuate in growth and spread, tailored treatments can take into account genetic variability, and they can better personalize treatments. An understanding of our immune system can even influence how we are enhanced genetically to assist in human immune system protection. As we seek to eliminate more and more threats towards human survival, it has become clear that biotechnology used in clinical medicine and immune system therapies has promising potential for the future of medicine. Firstly, I'd like to thank my colleague, Pam Matthews, for dedicating hours and hours and hours over the months to reading my lecture and fixing the stupidest of grammar mistakes, and spending hours with me going over my paper and helping me organize a mass of information. Every time I worked on my paper, I felt like I'd get more and more lost and just confused and unorganized, but she really helped me to organize my thoughts and put them down on paper. Secondly, I'd like to thank Jen Henderson, of course, for keeping me organized and up to date on the deadlines. She also proved very helpful when editing my paper, giving me helpful feedback, and she was also very supportive. I'd really like to thank my external advisor, William Black, for challenging me beyond my comfort zone and exposing me to the world of science, basically helping me start my scientific research and studies at the beginning of grade 8 for science fair. You've really gone above and beyond to help me complete this lecture and teach me the ropes of scientific writing and writing a scientific paper. Not only that, but you've also spent hours helping me fix my mistakes and showing me to edit and properly speak. You've really been an awesome mentor to me. Of course, I'd like to thank my family for supporting my scientific goals and providing me with lots of food for my writing, like lots of food. You helped immensely when it came to researching, editing, and helping me write, and just helping me understand and just read stuff that a first grader could understand. They helped to spark an endless curiosity, followed by my endless stream of questions. And I'd finally like to thank Dr. Hugh Black and Dr. John Hall for your inspiration and helpful feedback. You guys have been such a source of knowledge since I was, for a long time. And finally, I'd like to thank all of you guys for listening through this boring lecture.
Dr. Thompson, thank you for letting us attend your lecture. Learned so much, and I am so impressed with the depth that you went into this topic. It's really extraordinary, actually. So I'm going to hand it over to questions from the floor, starting with your advisors.
Colleague Thompson, very nice, very nice. I am so impressed. You pull off a really great teacher. That was not boring. But I'm not sure about the glasses, why you think that's funny. First questions are going to be from your advisors. Your first question is from Bill. We talked about a lot of things through your paper that weren't covered in your presentation. So one of the things we talked about was recent issues of COVID-19 and talking about vaccines. So you talked a little bit about vaccines. We did more in your paper really about it. But his question is, how long will it be realistically before we have a vaccine? And why do you think it's so difficult?
There's a short answer and a long answer. The short answer is between 12 to 18 months. And the reason why is because there's so many phases involved with making a vaccine. One, they actually need to make the vaccine, which can take a while. And then after that, they need to test patients, animals first, and then patients, and that can take a few months in itself. Each phase, they usually start with 20 to 50 people, and then they move on to a couple hundred, and then a couple thousand to test it and test its safety and effectiveness and how it affects different people, and to make sure there's no hidden variables and make sure that it doesn't come back and just even worse.
And this is a tricky vaccine too, isn't it? Because they're seeing different strains in different parts of the world, so there's no universal vaccine. Great. And his second question is, if it is too difficult to combat COVID-19 with a vaccine, what would you suggest? What do you think would be the next way to approach it?
There's two things that I see happening. One is something called herd immunity, which is when people who can't take the virus, people who have recovered from it, people who haven't had it yet, rely on them for safety. So to achieve that, it needs to be between 83 and 94% of the population that's immune to COVID. So everyone else who's not immune would rely on them to be safe from it, which I don't find being very safe, because the virus could always just mutate against that, and then you just have a big problem. Another thing I see happening is we just have to live around it for a couple months, and I don't know how long it's going to be, because things are always changing, and every day there's new information out there. So we're not 100% sure, but we might just have to live around it until we can just go back to being normal, I guess.
It's pretty exciting that the scientific community is sort of gathered together and sharing so much information about this to kind of try to fight it together, right?
Yeah.
Instead of competing against each other to find it first. Okay, this question is from Hugh. I have 2 questions. He sent out 15, so we're doing our best to narrow it down. He was asking, what do you think are the most deadly diseases that the world has faced in the last 50 years?
There's been a lot. One that comes to mind first is Ebola. That was a big one that hit a while ago. I didn't go too much into diseases, like all the different diseases that we've had through history. But there's a lot out there, like different strains, like SARS. We had different types of SARS. And there's also many viruses that people don't really talk about that harmed animals and that had massive effects on animals, like mad cow's disease and all these different diseases. And they may not seem very, not talked about a lot, but they still have had a massive effect on humanity.
And his other question is, what are the sources of these diseases? So how do you think these viruses, how does a virus just start?
There's actually many ways. It could start as a harmless virus, and then just by pure chance or somehow its DNA mutates, and then it can now attack certain people or animals or organisms. Then it starts attacking those organisms, and it can possibly mutate further, and then it's just not very pleasant. That's what first comes to mind.
So are there natural viruses that don't harm people?
There's many viruses that harm only humans and no animals, and then vice versa. I think there's lots of viruses that we don't need to really worry about unless they somehow mutate. There's probably thousands upon thousands of different viruses. Of course, because there's thousands, I don't really know all of them. But yeah.
That's your next master.
Yeah, virology.
Okay, we're going to move to questions from me. I had a little snicker because I think at the beginning of your lecture, Scott actually thought that you were playing it up and talking gobbledygook. Like at a point there, we kind of looked around like, is this guy for real? Is he talking this? And I can tell you that you are for real. You do know your stuff. We spent hours and hours pouring through every sentence of your work, making sure that we understood it. And I've been so challenged by your work because I had to do a lot of my own research. But of all the biotechnology techniques that you researched over the year, each one had a drawback, right? So each one you talk about how great it was and then say, oh, and there's these drawbacks. And so much difficulty getting past the first stages with these. You call them speed bumps in your work.
Yeah.
Which do you think is the most promising or the most likely to be implemented?
Some can be used, some are more effective against other certain diseases, but I definitely see CRISPR as being a big one. Though it's got many ethical questions, it's a lot more accurate, especially way more accurate than gene therapy, as gene therapy relies on viral vectors. CRISPR is very accurate and efficient, so I see CRISPR being a big one. And they just keep developing new methods of CRISPR. Like recently they developed CRISPR-Cas9 prime, and now that's a big new block of CRISPR that's really effective. So I think we're gonna just see more and more different types, and it's just gonna keep getting better.
Yeah, and there's a whole side of it that we talked about a little bit, the ethical issues of, your last slide showing the muscle men. There's that whole ethical issue around using it for good, right? So we have to hope that science keeps working on the disease side. And finally, my mom has MS. Do you think there's a cure out there for that disease? Because it's a tricky one, immune system, autoimmune. What do you think?
I think antibodies are monoclonal antibodies. They're a hope for MS. I think they're not 100% being used yet. They're still researching, and they have to develop a specific monoclonal antibody. I think they still have to do tests, but I think we might start seeing monoclonal antibodies used for MS and a lot of other diseases like rheumatoid arthritis. And I think it's just all a matter of time when we start seeing that.
I think of your work, that was the most interesting to me, too, to learn about. Thank you, Hunter.
Dr. Thompson, questions from the floor? And we'd like to start with Ms. Hunt.
Yes, Hunter, you're talking about antibiotic resistance. Three things that come to mind. So we're running out of antibiotics. We know we're running out of that. Quorum sensing is a relatively new thing, but phage therapy has been around for actually quite a long time, but it's not something that we ever really even hear about. And why isn't that more prevalent? Like, why aren't they using that?
So big drug companies, when they develop new drugs, they make a crap ton of money off of it because they can develop a patent on it. You can't really develop a patent on something that's natural. Like, it's like me developing a patent on J, which I don't really know what I'd use that for, but hey. But they can't make a lot of money off of that, and they're kind of all for the money because all of these developing these new therapies and treatments, it's really pricey. It's got a big price tag. So they kind of want something from it so that they can take that money and use it elsewhere, or else it's just going to be like using money somewhere, then they're not really going to be getting it back. But I do see that happening in some places that don't have a lot, like Georgia. I know that's a big, they're using phages a lot in Georgia, so that's a big thing. Yeah.
Ms. Stefani.
Thank you for a very enlightening lecture, Dr. Thompson.
You're so welcome, actually.
I was wondering if you could tell me about the process of finding new bacteria, and how is that different from finding new bacteria in phage?
So to find new bacteria or use different bacteria and collect them, first they gotta go like bacteria hunting, and to do that, they can use something called like an eye chip. It's kind of like bacteria bait. Like if you're trying to catch like a fish, you put like a worm on a line. But it's kind of like bacteria got something to munch on, so they're like, this is cool. What is this? So they're just like attracted to this medium, and the medium, it can change depending on like what you're searching for, I guess. But they can use an eye chip, and they basically just stick it in the dirt, just in places they think will be the dirtiest, I guess, because that's when you're going to find interesting bacteria. And I guess they can just start culturing it. They can put in petri dishes, and then they just start making more. And then I guess they can just start deciding what they can use bacteria for. And it'd actually be an interesting thing to look at, just genetically editing bacteria for antibiotics. That could be something interesting. What was the second part of your question? Did I answer that?
How is it different from finding new bacteriophage?
Phages, they basically just look in really dirty places like sewers and places where phages would be living. Kind of like, it's like the harsher environment. So like sewers and like places you don't really want to visit. Like, I can't think of it. Jay's locker. Sorry, Jay, I'm just targeting.
Okay, we're going to go to more questions from the floor. Adrian, go ahead.
Dr. Thompson, I feel like I was back in university and stumbled into the wrong lecture. I was too interested to leave, but a lot of it went over my head. But a question came to mind. It seems, from what I heard, that in the near future, there's a good chance that a lot of diseases will be able to be cured by CRISPR and other ways that we were talking about. Which brings up, as you mentioned, lots of ethical questions. Who do you feel should make those decisions?
Over whether we should be using...
Yeah, when to use it and who gets it.
I see a big disagreement happening between people who think we should be using it, people who don't. I think that could cause major political issues and problems. Like, this is some big stuff, and it's not as simple as, it's not super simple. So I think it might just be like prime ministers and presidents of specific countries that either enable it or not, or it might even be within states or provincials or provinces that decide. Or it might even just be people themselves can decide if they want to use it or not, and it might not everyone is going to walk away happy from this, but there's not a lot that you can do to really monitor it.
Do you see it as inevitable that it's going to be used?
I see it as inevitable that it's going to be coming up, and that it's going to be used, but I see a major conflict occurring because of it, which raises numerous ethical questions. So, yeah.
Okay, we have a question from the second finger.
Mr. Thomason, what an informative presentation. So obviously we've seen with the emergence of vaccines that there are certain people who are very opposed to the idea of getting vaccinated. Because biotechnology is such a complex thing to understand, do you think that this will be a sort of a fear of the unknown type of situation where the general population will have trouble coming to terms with this? And do you think that people will have trouble accepting this new practice?
Yeah, that's an interesting point you bring up. I think it's going to be like, I think it's going to start off, people are going to be unsure, but I think it's going to be, people are going to slowly get used to it. It's like people growing up with it are just going to see it as more normal, and then it'll be more and more normal for newer and newer generations. And I see it as, I think this generation is going to be more questionable towards it. Like, this is some powerful stuff. I'm not really sure if we should be using it or not, but I think the further we go, the more it's going to be accepted. But that's an interesting point, yeah.
We're going to go to our virtual audience. Start a question from Ted.
Right, from Ted Spear. Wow, you sound like a genetic scientist speaking to a medical convention, which is impressive. I wish you could go directly to university. Your explanation of the CRISPR array, among so many other explanations, was awesome. I hope you find the opportunity in high school or out to continue to pursue your obvious interest and ability in this area. Question. At the beginning of your presentation, I think you alluded to the possibility of human beings not dying. Do you think this is indeed a possibility, and do you think it would be a good thing?
So I think this comes down to the sustainability of this planet, because I know there's already kind of a lot of people. I think it comes down to, once we are approaching space travel more, I think we're going to need a couple more planets to have like 7 billion immortal people, because once immortality is around, then the population is just going to be booming. So I think we're going to need, I think they shouldn't allow it until we can actually sustain it. That's my personal thing, but I do think it's in our future that's going to be happening, or we just have like bodies that we go into after. We just transfer our consciousness and our brain into another body. That could be, that's many different avenues that could.
I think we are pretty close to being out of time. You've done an excellent job, Dr. Hunter, of answering questions. Not only was it a super informative presentation, but then we get to learn just as much in the next half hour. So thank you for that. We're going to have a 5-minute break and be back here for the Squirrels Village.
Jocelyn, don't you sit here?
Testing 1, 2, 3. Testing 1, 2, 3. Hello, hello. Keep talking. Oh, yeah, good one, Franny. Testing 1, 2, 3. Is that what you need me to just talk? Keep talking. Testing, testing. Testing 1, 2, 3. Hello. I'm really at this mic.
It's clipping, so it's not good to do that.
Do they possibly?
It's just because I'm trying to capture like all the questions as well, so it's not going to work. I'll just keep it here. It's going to have to be there.
And just vertical is the best.
It is, yeah. It's omnidirectional, so.
Testing 1, 2, 3. Testing 1, 2, 3. Check, check. Testing 1, 2, 3.
Delay.
Testing 1, 2, 3. Test.
It's a lot quieter.
Better.
Maybe I should put it back, but just lower the gain.
Stable enough?
Yeah, it should be fine.
Okay.
Do you want to try again?
Test. Testing 1, 2, 3. Check. Testing the new mic position. Testing 1, 2, 3. Hello, everybody. Testing Masterworks. Testing 1, 2, 3. Okay.
Tell me when you're ready. Hello everyone, welcome back to Masterworks. We're on our third presentation of the day. It's been an exciting week and it's going to continue all the way until Thursday, so please join us for all of those presentations. I have a few announcements to make before we start our third presentation, and this is about the first presenter, Mademoiselle Jada Madeline Scott, and I am happy to announce that she has met all the requirements of the Masterworks program. And to our second presenter of the day, Dr. H.S. Thompson. We were very enlightened by your presentation that we just saw, and I am happy to say that you have certainly met all the requirements of the Island Pacific School Masterworks program. So thank you everyone for coming. We have a unique Masterworks presentation this year. We have our grade 9s, advisors, and family of our presenters, a few grade 7s, thank you, and lots of people online because we are live streaming all the presentations through the week. So thank you for being here. Also, there's going to be a question period at the end. You can send your questions in if you are online to youtube@go.islandpacific.org. So this is a really amazing week for the grade 9s. They have worked so hard Throughout their years at IPS, I think they've seen presentations in grade 6 and grade 7 and grade 8 and thought, wow, what am I going to do it on? And that changes through the years, of course, but by last summer, they had to choose a topic that they thought would get them excited to learn about for an entire year. So this is a big decision. And once they have decided what they want to do, the grade 9s then get a faculty advisor, so one of the teachers at the school, to help mentor them through this, but they also get some external advisors. Now, these external advisors, we are hugely grateful to them because they make this program. They lend their expertise, they are professionals in their field, and they volunteer to help our 14 and 15-year-olds learn about the topic that they've chosen. So we really appreciate that, thank you. And today, lucky you get the gems of the research that they've done for an entire year. So this presentation will be a culmination. It is the simmering down, though, of a 15 to 40 page paper that they have produced and we have archived at the school. So if you want to read one of those, we've got lots of binders full of them. So I am very pleased to be the faculty advisor and also the Master of Ceremonies, and would like to welcome Miss Rose Willis to talk to us about her Masterworks decision.
Back in September, I couldn't decide on a Masterworks topic. And to be honest, I never really did. Hi, my name is Rose Willis, and welcome to my Masterworks on decision making, the best ways we can do so and the things that can hold us back. Oh, I forgot. In this presentation, I will be talking about making healthy decisions, the motivational value system, and how it can help us make better decisions, as well as the things that can hold us back from making these healthy decisions. As I mentioned, I was feeling very lost back in September. We had to make a decision about what we wanted to spend a year writing a paper on, and this seemed pretty huge to me. I've never been very good at making decisions, and it always took me a long time to choose anything. Most times I would close my eyes and point to a random option. As you can tell, this never really works very well. So when I finally came down to it, I said to myself, well, I'm pretty bad at making these decisions. Maybe I should figure out how to fix that. Thus, my Masterworks. When I had my first Masterworks meeting with my external advisors, I came to it thinking, okay, I'm bad at making decisions. I need a formula that will help me make good decisions that I can apply to anything. With the help of my advisors, we framed it completely differently. Instead of making bad decisions, I needed to figure out how I can make healthy decisions. And then I needed to figure out what even is a healthy decision. In this context, a healthy decision is one where the decision maker feels fully comfortable and confident with the decision that they made, and a choice that afterwards they didn't feel the need to question. Now, I want you all to think of a decision that you made recently. Any decision, big or small. Now really think about it. Close your eyes. And looking back at it, do you think that the option you chose was right? Or do you think maybe you should have gone a different way? If you were confident in your outcome, or the choice that you made, you probably used a healthy decision-making process. Having a decision-making process can be important to ensure confidence in your choices. An example of a decision-making process is the 7-step process. This is a fairly basic formula that's used for most decision-making in management. Ultimately, it's the same as any decision, but it's broken down into steps. Here's how it works. The first step is to identify the decision. This sounds pretty obvious because it is. It's just like setting the stage for the rest of the process. The next step is to gather relevant information about each option so that you can make an informed choice. The third step is to identify other possible options. You might find out while you're doing research that there are in fact multiple different choices, besides what you thought were your only ones. The next step is to weigh the outcomes of each choice. A pros and cons list is very helpful here. And finally, it's time to make the decision. Review all of the information that you collected in the first half of the process, and based on that, you choose an option. Once you've made your choice, you must take action and do whatever it is that you decided to do. Because what's the point of going through all of that for nothing? The final step of the 7-step process is to review your decision. Ask yourself if you're happy with the decision that you made, or if you're still wondering if you should have gone a different way. So as you can see, that's a lot of steps, 7 in fact, but this process is essentially a glorified way of making simple decisions. This method can be used on many decisions, from what to eat for lunch to where to go for university. And for simple decisions, the process can be simple as well. Any form of being thorough about making your choice will lead to a healthy decision. However, slowly analyzing a decision is not the only way to make a healthy decision. Healthy decisions come in many shapes and forms, and it all depends on the decision maker's goals, motivations, and values. As long as you make a decision depending on what you want, it'll most likely turn out for the better. How these healthy decisions are made depends on the person. Here are a few versions of making healthy decisions. We've discussed making a decision slowly and fully analyzing, and this can be called deliberation. But you can make fast decisions as well in a healthy way. This is assertive decision making. This is when a person is informed but hasn't necessarily done extensive research and comes to a conclusion using self-confidence and intuition. This is generally a healthy decision because if one is confident in their intuition or gut feeling, they won't feel the need to question their final choice after it's been made. So there's 2 ways of making healthy decisions, but you can make decisions fast or slow in an unhealthy way as well. Making decisions too slow or too fast can be called impulsive decision making. Usually impulsive decisions are made when the decision maker is fed up with their indecision and resorts to impulsive decision making in order to get it over with. Chances are, when making impulsive decisions, the decision maker isn't very confident in their choices, meaning that afterwards they might question their reasoning. On the other hand, making a decision too slow can be called decisional procrastination. And you can see that it's bolded in orange on the slide because I needed to remember to tell you all how much I love procrastinating and how this description completely describes me. Decisional procrastination is when a person feels overwhelmed by decision making, so they try to put it off for as long as possible. They believe if they stop thinking about the decision, they won't feel the stress that it can bring. They use procrastination as a temporary relief of stress, while in reality it's subconsciously snowballing, building in stress until they're faced with it at a more crucial time. Again, I love procrastinating. As I mentioned, healthy decision making is based on the decision maker's goals, motivations, and values. Most people know themselves enough to know what they find important and how they operate, but I got the chance to go one step further. One of my advisors, Andrea Raymond, introduced me to Total SDI, a self-assessment tool that helps people understand how their motives drive their behaviors and how these motives and behaviors change in different situations. Andrea uses this tool a lot to help teams at her company work better together. Part of Total SDI is the Motivational Value System, or MVS. MVS focuses on different groups that are categorized by their motivations and values. MVS can be visualized as an iceberg. The behaviors and personalities being the ice that shows above, and the motivations and values being what's underneath, supporting the ice above. This is the MVS triangle, and the 3 corners are the main categories: blue, red, and green. And in the middle is the hub category. Between the basic categories are different mixtures of the sections, and the main sections can be categorized by their focused motivations. Blue is people-oriented, red is performance-oriented, green is process-oriented, and hub is flexibility-oriented. There are about 5,000 spots that a person could be placed on the triangle, each with a different combination of the main motivations and values. Wherever a person is placed can describe their behaviors, motivations, and their values. As I discuss the different categories, I want you all to think about your own motivations and values and how you might fit into all of this. Which MVS do you resonate with? Now let's get into the sections. The first one is blue. This is the altruistic nurturing category. People who land in this section of the triangle are generally focused on the well-being of the ones around them and or who they're working with. Blues tend to find satisfaction in seeing the other person's benefit from the help that they provide, and they try to make sure that everyone is included and happy with the outcome when working with people towards a common goal. When in conflict, Blues are most likely to try their best to restore all harmony and end any conflict, and if this fails, they'll probably give in and learn to work around their failure. The next category is Red, assertive directing. What motivates Red is to achieve feelings of self-worth by providing the direction needed to achieve results and by being a successful winning leader to others. Reds value the setting of high performance standards, the exercise of power, and decisive action. When working with people towards a common goal, they will be focused entirely on the outcome of the project and getting it done as fast as possible and to perfection. They are motivated by performance. When Reds enter conflict, they'll most likely prefer to fight it out and are motivated to prevail against the other person. And then there is Green, the analytic autonomizing group. People who fall into this category are process-oriented. They are motivated by doing things methodically, logically, and thoughtfully, and by their own means. They get satisfaction from interpreting, and explaining, and discovering things based on analysis. When in conflict, Greens will resort to being cautious and want to avoid the issue for the time being. They'll try to solve the problem logically, and if this fails, they feel driven to retreat completely from the situation. Now, in the middle of the MBS triangle, there's a section called the hub, the flexible and coherent category. People who fit into this section find satisfaction by being flexible, responsive, and adaptable when relating to others. Hubs try to find common ground between all people to meet their individual needs and are willing to play whatever role is necessary to do so. They like to see all sides of the situation, and they have a desire to unite people together in order to reach a consensus. When in conflict, hubs will act accordingly to the situation in order to make sure that all sides are happy and can work together smoothly. They like to maintain peace and create inclusive working environments. So now that I've told you about all of these categories, which do you think you fall into? Take a second, and now raise your hand if you think you're Blue. And you're people-oriented. All right, a few of you. How about Reds, process-oriented? All right. And, that was performance-oriented. Whoops. And what about Greens, if you're process-oriented? And finally, Hubs. All right. Now, you might be thinking, Rose, what does this have to do with decision-making? And that's fair. Decision-making and indecision are what one might call a conflict of the mind. So, if we know how each MVS category might act in conflict, we can understand how one makes decisions when they are true to themselves, and how and why a person can struggle with indecision. By knowing what your motivational value system is, you can understand why you might feel indecisive, and therefore what the most productive way to move through conflict might be. Since you now have an idea of what your MVS is, try to think about how these descriptions could connect to your own decision making. I've already touched on how the different MVS categories act in conflict, so here's how they might handle decision making and indecision. Blues, when making decisions, will follow the path of least resistance, or the one with the least disturbance of other people. When they're indecisive, they'll try to avoid making decisions or put it off for as long as possible. However, they feel confident by basing their decisions on the people around them. For Reds, feeling indecisive is not a very common feeling because they don't like to hesitate much, and making a decision can feel quite simple to them. They usually go with the option that will help them achieve their goals, and a Red characteristic is self-confidence, so even if they have to make an unhealthy decision, they'll usually be confident that the outcome will be positive. Making a good decision for Reds is making a fast decision, often with the best immediate outcome. On the other hand, Greens, indecision is fairly common for Greens. Before they can make a decision, they need to think over every step and every possible event. so that they can confirm the decision will sustain a positive outcome in the future. When greens feel unable to make a decision, they will isolate themselves from other people and situations in order to think over the decision carefully before coming to a conclusion. For hubs, making a decision is about seeing all sides of the situation before coming to a conclusion. They like to incorporate and leave room for any new ideas, perspectives, or information to surface. This means that they might delay making a decision or commitment to anything in order to remain flexible. Ultimately, hubs want the most productive outcome for themselves and the people around them. Thanks to one of my external advisors, Andrea, I was able to take an SDI assessment and learn about my own MVS, and I discovered that I am a hub. You can see there, that cute little black dot, that's me. I'm right in the center of the triangle, but I'm also a little bit towards the green-blue side. The motivations of a hub are when working around people, they want to make sure that everyone is getting along and working well together. And if this isn't the case, a hub is prepared to change their own opinions and or goals in order to have the rest of the group function smoothly. This is something I feel like I do a lot of. I hate it when people are fighting, so I'll usually end up trying to be the peacekeeper. Also, when I'm making decisions, I feel like I need to see all sides of the situation before I can make my choice. And sometimes this can feel like I'm constantly spinning between my options, trying to see everything. This is where Andrea was teaching me about SDI and MBS, and as you can see by the books and papers all over the table, I had a lot to learn. Anyways, the motivational value system does a great job of breaking down these different groups based on behaviors, motivations, and values, but all it really is about knowing yourself, and you don't really need any tests for that. However, by knowing about your motivational value system, you can understand the motivations behind your values and behaviors and how it all relates to individual decision making. And that will set you up to confidently find a process that you can rely on. Now, we've discussed making healthy decisions. We've discussed making healthy decisions, but what are the things that can hold us back from doing so? Oftentimes, aspects of our general mental health can make it a lot harder to make good decisions. This includes depression, anxiety, environmental stress, and neurodevelopmental factors that can impact an individual's capacity to make good decisions. The first of which I will be talking about is depression. Depression is a mood disorder that causes a constant and persistent feeling of sadness and loss of interest. It affects how you think, feel, and behave, and can be caused by an imbalance of chemicals or hormones in the brain. People who suffer from depression make decisions completely differently compared to a mentally healthy person. This is because depression can change a person's motives. They will make decisions depending on the safest outcome, and they will stay away from any risky options as much as possible to reduce feelings of anxiety later on. Some people who suffer from depression will also try to avoid decision making in general because they want to try to minimize any regret they might feel in the future. This is, in a way, accepting the default option. If a decision is made that led to a negative outcome, someone with depression will feel much more regret if it was their decision than if it was out of their hands. This is because if it was their decision, it means that it was their fault. The next factor that could affect a person while making decisions would be anxiety. There can be many different variations of anxiety disorders, but all can be characterized by a feeling of worry, fear, and sometimes leading to physical symptoms of fast heart rate, shortness of breath, and shakiness. The word anxiety can be used as an umbrella term in this context to understand the direct effects that it has on decision making. When a person suffers from anxiety, their brain can be easily overwhelmed and overloaded by simple things and information. This is because anxiety overrules the brain's ability to ignore mostly irrelevant distractions, such as the environment or thoughts and worries. This overload causes the brain to shut down specific areas of itself, specifically the prefrontal cortex, making it even harder to screen out irrelevant information. The prefrontal cortex is where rational decisions get made, executive functioning, regulating emotions, risk and reward calculations, and long-term planning happens. When anxiety causes the brain to turn off parts of the prefrontal cortex, it directly affects our ability to make good and healthy decisions. Similar to people suffering from depression, a person with anxiety will also do their best to avoid risk. when making decisions. They want to minimize any increase in feelings of anxiety. Depression and anxiety are both factors that affect our decision making from within, but decision making can also be affected from our environment surrounding us. Not only can our environment directly influence our decision making, but it can also be a notable source of many mental health disorders. Environment, in this case, refers to the people around us and whether or not they create a positive space to live and think in. If the people around us have different values, motivations, or for us, this dissonance can cause stress in our own decision making. It can be very difficult to make decisions that agree best with one's own goals, motivations, and expectations when there's a parent or peer who has a different and perhaps more vocal expectation for us. Even though the decision maker might not agree with the parent's expectations for them, their opinions could be overruled by the parent, and the decision maker could be forced to choose an option that they might not otherwise have chosen. Decisions will almost always turn out best if they are made according to the decision maker's goals, motivations, and expectations. Finally, decision making can be extra difficult for teens and young adults because their brains are still developing. Decision making, as we know, relies on the prefrontal cortex, which controls rational thinking. Things like depression and anxiety inhibit the prefrontal cortex's ability to make good decisions, but also the prefrontal cortex is still developing until the age of 25, meaning that decision making is extra hard for teens and young adults. While the prefrontal cortex is still developing in teenage brains, the limbic system develops much faster. The limbic system is the part of the brain that controls emotional responses and will often take over when decisions need to be made before the prefrontal cortex is fully developed. The relationship between the prefrontal cortex and the limbic system explains why teens often revert to making rushed decisions in emotionally charged situations. The other reason that teens find it often harder to make decisions is that they're still developing their identities. It can even take a lifetime for someone to fully understand who they are in the world and who they are as a person and how they fit into the world. Identity is the base of a person's goals, motivations, and values, all of which are key in decision making. Therefore, if a person has yet to find their true identity, it will be hard to make decisions that benefit them in the best way possible. So, now I've told you about making healthy decisions, decision making by your motivational value system, and the things that can hold you back from making healthy decisions. But what it all boils down to is about knowing yourself so that you can make decisions in the best way possible according to you. After I learned about all of this, I felt like I needed to put it into action somehow. And so that's what I did. As you all know, I'm finishing my last year at IPS. Therefore, in about 2 months, I'll be going to a new high school. Before this year, I had always planned on going to Rockridge Secondary School, a high school that I'm in the catchment for and generally the accepted choice for most Bowen kids. But then around October, some new information came up, and I realized that I had more of a decision to make. I had to decide between going to Rockridge and West Van Secondary. This decision was mainly based on the social aspects of each choice, and I think this was partly because I'm a hub, and so my mental state is constantly dependent on my social environment. As you know, I'm quite decisionally challenged, so I knew this decision wouldn't come easy. I did my best to simplify my choices and stick it through the 7-step process. And this mainly worked, but as I'm a hub, I like to see all sides of the situation. So I needed some more information before I could decide. I did some research and gathered information by talking to peers who went to each school. I also attended tours of Rockridge and West Van. While I was thinking about this decision, it felt like I was constantly swaying back and forth between my options. One day I would be certain about my choice, and the next I would be questioning my reasoning from the day before. However, finally, after I felt like I knew enough about both schools, I was able to make my decision, and I'm happy to say that in the fall I will be attending Rockridge Secondary. I'm confident in this choice because I used a healthy decision-making process while also taking into account my MVS and my mental state. To conclude, everybody wants to make healthy decisions. A healthy decision is one where the decision maker is confident and comfortable in their choice because their process was healthy according to how they function. How they function depends on their decision making or their motivational value system. By knowing about your motivational value system and yourself, you can understand the motivations behind your choices and the reasons that you might have a difficult time making decisions. Your MBS can sometimes make it a lot harder to make decisions, but there are other reasons behind this as well. Mental health and your environment play huge roles in decision making, and knowing about all of these things and about yourself makes you better prepared to make healthy decisions. Learn about my own. And Carla for the multitude of sources and websites and being an incredible bank of knowledge when it came to mental health in decision making. Thank you both so much for being so very excited about my project and for helping me with my paper structure. Also, thank you for taking time out of your own busy lives to read and edit my 30-page paper multiple times. I would also like to thank my family for supporting me throughout this process and throughout my life. Thank you to my classmates as well for providing comic relief in stressful times. And finally, thank all of you for listening to my presentation and giving me a place to share my work. I hope in some way I was able to help you understand the decisions that you might struggle with personally. So thank you. I can imagine that people change as they develop and as they grow, and as they find their identities. I feel like I'll probably always be a hub. I feel like that's kind of a main point of my personality. But I think it's definitely possible for people to change.
So next question. Are you drawn to blue, green, or red and would like to be more that way? And do you think that you can actually sort of train yourself to be more assertive, more nurturing, caring, or more logical, analytical?
Maybe Andrea has more insights on this, but I think that you could probably, you could definitely train yourself to be like more like one MVS. I think sort of the one that you're originally associated with is always going to have underlying like effects on your personality, but I think it's possible to be more drawn to another MBS and to change your motivations. I like being a hub. I like being flexible, so I don't think I would change myself, but
cool.
Yeah.
Okay, I'm going to turn it over to questions from the external advisor, starting with Andrea Raymond.
Rose, I was thinking about your multiple perspectives that you have as somebody who sits in the middle, who can really see the reds and the blues and the greens and all the colors in between. And I'm wondering whether understanding a little bit more about yourself has informed your art.
Well, that's a good question. I really haven't thought about that. I think in some ways it has. I think knowing about myself has affected other aspects of my life. Like I know more about making decisions for sure, and I know how I function a lot better. I'm not sure if it has affected my art. I haven't noticed anything, but I think now that you brought it up, it might.
Good.
So over to House Group, it's another external advisor.
Rose, thanks again. This was just wonderful to see you piece everything so well. But I wanted to know if there was something that initially you perceived as a weakness, or maybe something about your character that you felt challenged by, but then through this process that you realized maybe that wasn't so much of a weakness, but maybe it can become more of a strength for me.
Yeah, I think definitely. Well, I started out this year thinking I'm bad at making decisions. I procrastinate a lot, and I thought that was what I would call a bad decision, but I think I've learned that procrastinating, it comes from a different place. Like I procrastinate so that I can, because I'm a hub, I procrastinate so that I can see all sides of the situation just in case anything new comes up. And I think sometimes that's a good thing. I mean, it's a lot harder to get things done, but it's good that I know that I can, take time to incorporate anything that might arise.
So we're going to move to questions from the floor. Does anyone in the audience have a question? That's Jan. Can you please stand up?
Great presentation, Mrs. Willis. I enjoyed it very much. I was just wondering, what do you think has been the biggest decision that you've ever made so far in your life? And what were kind of the deciding factors, and if you think it's a healthy or unhealthy decision?
Well, I don't know what the biggest decision I've made would be, but definitely a large decision recently was my high school choice that I touched on. And I think in the end it was a healthy decision. I started thinking about it way before. I had really gone deep into my Masterworks, so I didn't really know much about it in the beginning, but I think that, in the end, since I waited so long, it became a good decision because I was waiting for everything to come together. And it did. And I think having knowledge about the 7-step process and making healthy decisions, that helped me a lot because I could sort of set it up to be good.
Questions from the floor? Ms. Madeline.
Congratulations, Ms. Lewis. I just wanted to ask that you say that impulsive decisions are unhealthy kind of version of decision making. So I was just wondering how you actually just like the whole 7-step process actually go. Do you use that process in small stuff like shopping?
Well, that's a good question. The 7-step process is basically any decision that you make in some way or form. You probably go through it. It's just how in depth you are with it. I think with impulsive decisions, it's a lot because a lot of the time the decision maker doesn't go through that process. It can turn out unhealthy. I mean, there's always a little bit of wiggle room because, sometimes things turn out for the better, but I think it just depends on the person.
So I think your mom has a question about the decisions you had to make just before this.
Right?
Well, a better question actually. How do you see your placement in your family configuration as a factor in how you make decisions?
Well, that's a good question. I am the youngest, and I think sometimes that plays into it. I feel like sometimes I don't always have a lot of weight in the decision making, but I feel like our family specifically isn't very good at making decisions as well. So I think, sometimes it feels like I'm not quite heard because I'm the youngest. But since I'm flexible as a